Hello! Today we're going to be talking about the role of tRNA in translation of genetic material to functional proteins. There are three stages of genetic material. The first is DNA, a double helix of nucleotide base pairs. In order to get to the final stage, which is the production of the protein, DNA must be transcribed into single-stranded RNA, the second stage. RNA is a copy of a coding strand of DNA. The only difference is that rather than pairing thymine with adenine, RNA uses uracil to pair with adenine. There are many types of RNA, but today we're going to be looking at mRNA, rRNA, and tRNA. When messenger RNA, or mRNA, is created, it carries the message of the gene code to the ribosome, where it is translated into an amino acid chain, which eventually forms a complete protein. The function of rRNA is to combine with other proteins to form these ribosomes, which again are the site of translation. The last of the RNA we will look at is transport RNA, tRNA, which houses a specific anticodon, or set of 3 bases in a particular location on the tRNA molecule. The anticodon sequences serve 2 roles. One, it charges the tRNA with a particular amino acid. The second role of the anticodon is to pair with codons in the mRNA. A codon is simply a sequence of 3 bases that, in mRNA, complement anticodons on the tRNA, bringing both the tRNA and the attached amino acid to the translation site, the ribosome. Then, part of the rRNA of the ribosome acts as a ribozyme, creating peptide bonds linking together each amino acid that is transported by tRNA to the ribosome. Codons and their associated amino acids have been established by researchers in the past, and we will use this dictionary as a reference. The attachment of amino acids to the chain thus follows the sequence from the mRNA, which originated in the DNA. Francis Crick referred to his proposed function of tRNA as adapter hypothesis because tRNA serves as an adapter between amino acid language and nucleotide language. Think about it this way. Sometimes, when you go to another country, the outlets there look different, and if you tried to plug in your American-made phone charger into their outlet, it would not work, both physically and electrically. You need an adapter. The adapter allows you to plug your American phone charger into it, and then it plugs into the foreign outlet, allowing you to receive electricity from the outlet and charging your phone. Now, imagine your phone charger is the amino acid language, and the foreign outlet is the mRNA nucleotide language. You will not be able to transfer electricity, or in the case of translation, genetic information, without some sort of adapter. tRNA plays the adapter role in the transcription process, bringing associated amino acids to the ribosome via complementary base pairing between the anticodon and the codon of mRNA. It is because the anticodon on tRNA is associated with a particular amino acids and a particular codon sequence originating from DNA that the language can be changed from nucleotides to amino acids, forming the genetic product of a particular protein. When tRNA is first transcribed from the genome, it must go through a period of processing during which cleavage, splicing, base addition, and base modification occurs, creating a secondary structure, which positions the anticodon in the proper physical location to pair with mRNA, as well as charge the tRNA with the amino acid. If the secondary structure is not formed, one or both of these cannot occur as the tRNA is misshaped. If the tRNA is non-functional, it cannot add amino acids to the protein chain based on the mRNA sequence, and translation as a whole would not occur. This would render DNA useless, and the cell likely would not survive because it cannot create proteins vital to survival.